So I want to start off by introducing our guest speaker here today. It's Dr. Lussier. He is a professor and chair of the bioinformatics department here at the University of Utah. He's been here for somewhere around 9 months, we were just discussing. So he received his medical degree from the University of Sherbrooke and then did a postdoctorate in biomedical informatics at Columbia University. He has had a fairly extensive impact on the field with over 100 presentations and 100 publications, as well as appointments at Columbia, University of Chicago, University of Illinois at Chicago, and most recently University of Arizona, where he was the Associate Vice President for the Information Sciences and the Chief Knowledge Officer there. He's had involvement both in the private sector, but also in education throughout his career. And please welcome me in joining Dr. Lussier.
Thank you. Can you hear me? Good. So I won't try to pontificate in radiology. My tenure was in clinical informatics and genomic medicine, precision medicine. But I'd like to share the lesson we've learned in a domain that's much easier than imaging. Please don't tell that to my peers. But precision medicine has crisp data that's well annotated, while in imaging, as you know, the features varied from one person to the other, since we're all pretty unique physically. So it's more complicated to go from machine learning into imaging than it is to the genome. And most importantly, it's really sharing with you a vision of how we're agents of the future. We're privileged to be in the academia. Some of us could be in the industry, in R&D. And the future is the one we envision. And some of us can really set the pace and determine what we'd like the future to be like. So if, like we heard the naive journalists have been telling us for over 10 years that pathologists and radiologists studying images will be out of a job soon, since machine learning is coming in and will commoditize these jobs. Well, that will be the future if we don't, show them it's different. And before starting, let's just think of this for a minute. I remember 10 years ago, or a bit longer, the mammographies. I remember that it was pretty challenging. There was this idea that perhaps we're creating more cancers between age 40 and 50, and we should just do the mammography by age 50 if a woman didn't have prior family risk or personal risk of cancer. And all of that depends on the quantity of radiation. If machine learning requires only 1/10 of the radiation, though it's not human interpretable, but it's as accurate as we are in reading a mammography, it changes entirely the strategy we could have. We could have a two-stage system where the first stage is machine read. Maybe you could start at 25, right? You can think of that for the pancreatic cancer that we can't detect early enough right now. Could we work with low dose or less invasive imaging technologies that are machine read? But you still could increase actually the volume of positives that need to be verified by human beings through conventional methods. And that changes entirely what we'd call the break-even point, right? You could actually need more pathologists and radiologists. So that's the challenge we have right now. Do we want to envision the naive future that journalists are telling us we're going to aim for, or really execute on a future that's more interesting for the population, which is not doing more of the same with machine learning, but empowering physicians to do better. Okay, can't change. Okay, now it's working. I don't have any conflicts, I think, but these are where my students, at the bottom are where my students are now practicing, and on the top were former conflicts or current nonprofit boards in which I still sit in the intellectual property of several institutions. Again, let's try to imagine if we could generalize insight from a case report. It's really hard to do right now. Could we investigate very large data sets and come up with hypotheses For example, new radiologic signs. Some 30 years ago, when I was a resident, I started a company, I was co-founder of a company where we wanted an AI to reside on the computer. There was no electronic form of data dictionary that made sense for the computer. The language of medicine was not understood by computers. So we created a network of relationship of diagnoses. They're underpinning procedures, etc. And these type of data structure, I'll show you more of these later, allowed an AI to reside there. What we discovered while doing that is you could actually unveil from large data set a lot of new knowledge in medicine that eluded clinicians. So there are principles of medicine that are recapitulated by these technologies, and that confirms that the systems are working, but they also unveil new facts and new knowledge that we can use. But we need to train them into that in a non-trivial way. So this is the plan. I'm going to hope to discuss the problems with current machine learning approaches. Again, I don't want to pontificate in imaging. I know that you're using a lot of machine learning right now, but I'm also aware of a lot of history of expert system machine learning that comes from informatics. Some of our luminaries have pioneered the field of expert system, like Ted Shortliff, which was reasoning with logic and reasoning with uncertainty at the same time. That dates back from the mid-70s from Stanford, and different from the black boxes we see, like neural networks and random forests, and probably the machine learning that are working on your apparatus right now. These were able to explain the decision-making, and it was shown in a clinical trial that it could beat infectious disease specialists in treatment decisions, but it could also explain the treatment. Now, we're speaking of 75 to 80, right? And we're 30 years later, 35 years later. How many of your AIs are explaining their decisions to you? Raise the hand. Tell me which ones are. We forgot people don't read literature. We reinvent the wheel back to the 50s. In the 1950s, the mathematics that support neural networks were developed. The statistics of Bayesian statistics were dated even further back, and the inference engines of inductive, deductive reasoning with Prolog and Lisp and other languages like that are different language than the current programming language that you're familiar with, were able to reason with logic. All that dates back from the 50s or before, but they're all different fields. And there's someone in the field of informatics that created the Maxwell equations that, like for electricity, that brought magnetism, electricity under the same set of equation, created that for artificial intelligence. It was done under the mentorship of luminaries of those that developed the previous technologies in the 50s. All I'm saying is that there's a lot of reason for which black boxes may be detrimental to patients, and I'll get into that. So let's go into the detail. This is just a newer paper, but you've seen many papers of convoluted neural networks that feed into one another. And I'd like to show you a bit, some are better illustrated in this. I couldn't find one, but there are many scales of information. So raw data starts from the top, and then there's a scale feature that's learned, and another scale, and another scale. As these scales of features are learned, they could be learned like this model here in a very naive way that cannot be human interpretable. But we know 4,000 or 5,000 signs of radiology. Why don't we have the systems learn them so that at a certain scale that corresponds at the scale of the radiologic signs, it speaks the language of physicians as well. Not just the diagnoses at the end like we're doing right now in this model. How many of you are familiar with the wolf versus dog paradigm in machine learning? Yes, a few. Well, okay, so it's worth telling the story. So Google and Microsoft provide machine learning tools for free, and they've mined very large corpus because in the previous model, if you don't have enough images to train early on, you can borrow features from these large training set. And here's the biases you bring in. There was this very good model. You borrow it, and you can find what's a wolf and what's a dog. And it's worked so well on thousands, tens of thousands of imaging for years. Then someone tried to figure out, what is it reasoning on? What is it reasoning on snow and colors, not at all the features of a dog or the features of a wolf. Think of snow and colors as race, ethnicity, gender, and the dog as being a diagnosis, or the wolf as being a severe diagnosis and a milder one. So now we're having a system that's just looking at the color of the skin of people and where they came from by ancestry, and it's looking into the frequency of one having a disease versus another one, not at all the clinical facts. We call that racism, right? and it's an EDI problem. So the neural networks that are trying to decide a decision and can't explain themselves are at risk of all these biases. Now this is just, I'm not sure how many of you are familiar with the systematized nomenclature of medicine. SNOMED, okay, it's been used by Kaiser Permanente for 20 years. It's owned by an international organization, but earlier it was owned by the American College of Pathology. And I was on the board when we made it electronic. So that's 10 years after I did that startup. We decided we needed that terminology we could share, but it's not just a data terminology, it's a network of relationship of what a disease is defined as an abnormal function or structure with or without an etiology. Procedures are defined with the instrumentation. So you have really a periodical table of elements and the chemistry that works over it to make molecules of facts. It contains 350,000 concepts of medicine. It's got 7 types of anatomy, embryologic. So you could ask, coding our data sets of the hospital into SNOMED and with many others, and we did that. You can ask, what are the cancers that arise from the mesoderm or the ectoderm? So tell me which physicians can do that on top of their head. But we could ask that, and you could look into the frequency, and it could change maybe by region, and it does. And what are the molecules that are more into mesodermic cancers that arise from that embryologic tissue? There are 7 types of anatomy, even the regional anatomy. You could ask, what are the molecules that arise from, that lead to cancers that are of the thoracic region or the abdominal region or the retroperitoneal region. You can do a lot of different thinking. Though physicians know these anatomies, reasoning in the high throughput with these anatomies is not the way we've conceived our subspecialties. We've conceived them by organ, systems, not even at the tissue level. And so this language now is organized as a network, and there are statistics and methodologies that work over these networks that can generate new knowledge as well. This is just to illustrate what these networks look like at the biomolecular level, and you've seen that, but at the clinical level to the left, it gives you an idea of the type of semantics that relate concepts, and the type of maths that work over that is subsumption, and that dates back from Aristotle and Plato, reasoning with logic. That's another view of what SNOMED would look like when you want to code an H. pylori-associated hemorrhagic gastric ulcer. And you see the code, and you can kind of see it's like a molecule. You built it as a molecule, right? The concept and the diagnosis contains 3 components that come from the anatomy, which is called morphology and topography. So the abnormal anatomy and the anatomy. But that's how it works, and the computer can understand that. So, so there's an opportunity to conceive and design convoluted neural networks so that their scales of features that are being learned coincide with the scale of biology we know. And if we do that, it's likely to find features, recapitulate features we already know, and disclose new features we didn't know about at that scale. And that means new radiologic science that could be microscience that support the macro science that radiologists are probably more familiar with. So we call these intermediate phenotypes or mesophenotypes. And I'm speaking of a spherical elephant analogy because, you know, if you ask a physicist, how do you measure the surface of an elephant? Well, the minimum surface would be like a sphere around the elephant, right? So you take the diameter of an elephant and the spherical elephant, and then the maximum surface is a bit more difficult to assess, but at least you have an estimate of the minimum one. There are analogies in the omics space, which, as I said, is easier to understand than the imaging. And we've learned meso-intermediate phenotypes that are very meaningful to clinicians, much more than the molecules. And I'll show you how that works and how we operate that. And it's an analog, but some of these technologies are immediately portable to radiology. And again, each person is unique. And in the genome space, we've got this advantage that though each of our sequences are unique, there are regions of sequences called loci for which we've assigned genes, or pseudogenes, or other function, and we can then try to interpret and create a matrix of commonality between individuals and know when a mutation at a certain site affects which region of which gene, and there's enough similarity between these regions, between individuals, that we now have a crisp understanding of the genome that's comparable from one person to the other. In imaging, that's probably several scales of your convoluted neural network. You need to learn the micro edges of what's going on, and eventually, once the features, if they're designed to be understood by humans, correspond to tissues and organs and systems, that makes a lot of sense, right? But if they're designed by computers and the number of, it's arbitrary how many layers you put in your neural network, and you decide to use layers that are nonsensical to human beings and to the structure of the biology, I don't even think it's a good design for the learning system. There's a good reason for which the surgeons will dissect according to certain planes and certain tissue, right? It's the same thing when you do imaging reconstruction. So it makes sense also to have the neural network designed so that the scales work with these known biological facts. So in terms of interpretable machine learning and explainable AI, who's read about IML or EAI before? Okay, so that's how the community calls these aspects. This is just 3 editorials or 3 articles that describe it. There are dozens and dozens that make a case for, go back to the basis, the basics. 35 years ago, it was unacceptable for clinicians to have an uninterpretable diagnosis, and expert systems were designed so they could reason with logic and uncertainty and explain what they were doing. So how can we do that with the new forms of machine learning that are more powerful, like the convoluted networks? And you can see, as I pointed out, Ted Shortlift during his PhD under, I'm trying to remember who was his mentor that had invented the previous forms of AI, but these were the characteristic that's important to them, but black boxes are unacceptable. Zach Kouane says the same thing in the New England Journal. Zach is from Harvard. And then Jason Moore, formerly from U of Penn, again, the same thing. These are all colleagues. I've published with the 2 first ones, well, the three of them, actually. So these are just colleagues. That's known facts in the community. And we're trying to share that with other communities because obviously there's a big excitement and investment right now. I'd like to give you an idea of size requirement in omics. And again, I don't know what that means in your field, but there's got to be a corollary. At the DNA level, a genome-wide association study works on over a million features, a million SNPs. And it takes 4,000 subjects in the discovery phase and about as many in the validations phase to find signals that have an effect size of about 1%, 0.5% in inheritability. So it's a very low signal to noise, and it's a very low effect size, and that's why it takes so many patients. When you have a variant and you want to know if it's important, if it's a new mutation or a non-mutation, and what's the frequency of that mutation in an individual, we have reference genomes. Now they're at 100,000, but there was a time where there were 1,000 and people felt it was sufficient. Below that, probably not. So at one time, someone has to construct a reference genome. So it does cost the cost of 1,000 sequences. And again, if you look at the SNPs, it's probably 20 million in Caucasian. If you look at the base pairs, you're at 2.5 billion features. But there's a reference, right? So you can know if the frequency is below 1 in 1,000 or 1 in 10,000, your reference. As soon as you go into the dynamic gene products, proteins, metabolites, messenger RNAs, our team is the one that proposed that reference transcriptomes or proteomes are nonsensical. We're our own reference. You all know of twins you may have imaged or you may have read about, and because they ate and lived in different ways, they actually don't have the same phenotype. And some are diseased, some are not, and they're exact twins, right? So being our own control is probably the best strategy. And you can see at the bottom of the table, how many patients does it take to be their own control and how many samples, right? Just one. There are new statistics for that. We pioneered those. And you can get p-values and effect size at the pathway level of what's going on in one patient. It's not generalizable, but to generalize, instead of requiring, as you can see, by conventional general linear model, the t-test or these type of statistics, 500 or 100 patients, you need only 10. And you cannot generalize. That's a dimension reduction that's substantial for clinical trials. What would have been a pilot study for someone can now be published with validation. What was inaccessible for a junior faculty member, and they had, unfortunately, think of junior faculty members today that want to do clinical trials. They have to use frequent diseases, and they've been overstudied by senior people, and the rare ones require very large grants and multi-center accrual because of a rate-limiting aspect, and therefore they're stuck, right? Because it's hard to have a lot of novelty into well-studied diseases. You microstratify them, you get into the same problem as rare and infrequent disease. So what we're trying to propose are new strategies to enable the next generation to go ahead and study stuff we didn't study. Now, I'll have a slide later. How many of us are clinicians here? The majority, I guess, or at least have the room. We've all studied probably 40 to 65,000 diseases in med school. They say we learn the language of 4 or 5 different languages when we do med school. But do you know how many of those we treat? There's only 4,300 distinct compounds that are FDA approved. Of these 4,300, there's only 2,000 indications. And many of these are symptoms. We treat less than 4% of diseases. The other ones are infrequent, but in pediatric clinics, they still correspond, infrequent disease are still 30% of the visits, for which we have no drugs, no treatment. They don't fit the blockbuster model, where we need a very large market to support the development of a phase 3 clinical trial that costs $350 million. So if you reduce the size of clinical trials by 5-fold, it's not to make the drug cheaper for frequent diseases, it's to hopefully address, if it's a pyramid of frequency, the next group of 15,000 diseases and move out of treating 2,000, what we're doing. So I'm saying that because we're all trained with blinders. And I'm the hope after this presentation, you peel off some of your blinders from our training. We don't realize it till we look at the facts. How many of you realize that we treat so few diseases? They're very frequent, right? It's always diabetes, hypertension, very frequent diseases, strokes. And in terms of the other problem with microstratification when we do single subject studies is that one of the other blinders we have societally is to try to assign people to a race. It is absolutely nonsensical to compare an African American to another African American as groups. Nowadays, it's modern science. You need to put into your grants a bit of genetics to look at the admixture of their ancestry. Some of them could be 40% Caucasian, some 20%, but in Africa, 2 villages may be more different than European and Asian. Out of Africa, there's only 20% of the human variation at the genetic level. 80% is in Africa. 2 villages in Africa, fundamentally, I hope you remember that coming out of here if you do a clinical trial, have more genetic differences than European and Asians, right? So please keep that in mind. And when you start creating your clinical trials that become so small that they're microstratified with the new statistics we have, and we know that, we're funded many grants, and we experience that, it's very, very hard to pair Africans. We need to have the genetics as well, and it's very difficult to address. So that's a covariate that's very difficult to mitigate. But in a single subject study, where you take the tissue of someone, like the blood, you do an ex vivo assay, like exposure to a certain drug, to estimate their response, and we've done that. It's actually very good, because you've controlled in isogenic form, as if you were in a cellular model or an animal model like a mouse. And what I didn't tell you in the previous table is that while it takes 500 people to do a classifier at the transcriptome, or 100 people to have reproducible mechanism between two groups of phenotypes from the peripheral blood, for example, it takes only 3 mouse per group, right? Because they're isogenic, they're clones. And that's why I find always funny when someone generalizes from one strain of mice a cancer treatment to do a publication. Because sure, maybe this treatment works in Yves or Satoshi, but it works in no one else, right? So they need to try it in the different different mouse models, and otherwise it's not generalizable even in mice. So 20 years ago, there was this publication of the human genome paper, and the question was, do we do single markers or do we move to multi-gene mechanism? Unfortunately, people have not moved to multi-gene mechanism. We spent billions of dollars in biomarker discovery, not just single molecule, probably other single biomarker in other field. You may remember that maybe in radiology, but it's been very unproductive. Single molecule biomarker, you can see the period of 2012 to 2017. It's hard to get these facts, but in cancer, there's only 5 discoveries. Now, I think Einstein said something about applying the same rule over and over again and expecting a new result, right? I think we have enough exposure here to say we need to change the strategy. And single biomolecules assume that they're concordantly altered across patients. Well, of a group that has a case in the control, that's a perfect model for Mendelian disease. If we were subjects of Mendelian disease exclusively, we'd have 25,000 diseases. But I told you we know 65,000 diseases and 25,000 genes. So there's a problem there, right? So it's multi-gene diseases are not modeled properly. Yet, that's what our peers are funded. You know what it reminds me of? It reminds me of the MRI that couldn't get funded at the NIH, that won the Nobel laureate, because there are physicists that knew at the NIH in the review board that this would never work, right? They knew better. Another example here. Hemophilias were the first genetic disease that were discovered because the royal family in Great Britain suffered from that, so they paid genetics on that, and well, they didn't know it was genetics, but studies on that in the mid-1800s. Every molecule here, a loss of function, will yield a prolonged bleeding time at the bottom of the pathway. If you have loss of function of molecules on both pathways, the extrinsic and the intrinsic, it's embryologically lethal, so you don't have human beings that have that. But the prolonged bleeding time, that intermediate mesophenotype, it can be found also in alcoholic with chronic cirrhosis, or anybody else that's got a chronic cirrhosis. In this case, there's absolutely 0 mutation. It's an acquired trait, and it's acquired by a reduction of production of 4 proteins in red. And the summative effect of 4 protein at low expression gives the same prolonged bleeding time, the same intermediate phenotype. Again, let's think back in the radiology what that means, right? When you're studying different diseases, there are intermediate phenotypes that are common. Your machine learning algorithms can learn from that the language at different scales of imaging that could talk back to you, and some of these will recapitulate your known radiologic signs, and some will be new ones. So we're proposing multi-gene biomarkers and genome by environment assays, and I'll get more into the detail of that. So in terms of clinical trial design, this is a generation. The third industrial revolution was a digital revolution with machine learning, And we could do it in a very silly way and apply it like the second industrial revolution, which was the travail à la chaîne in French. I'm trying to remember how that's said in English. Line work, assembly line work. So everybody works on their piece of it, and we just do an assembly line and do their clinical trial like that. But the fourth industrial revolution was really, radiology was a pioneer of that in medicine. CT scans, MRI, you had to redesign sensors, the hardware. You had to rethink the maths. I mean, it's an Austrian that developed the maths in the early 1900, 1920 for the CT scan, right? It wasn't applied for the CT scan. It was simply maths that lay out there. And then it's put together with the biology and the knowledge of medicine. That's the fourth industrial revolution. It's to think like a system. And if we want to redesign clinical trials, and you'll see in genomic space how we have to redesign clinical trials in order to take advantage of single subject studies, we need 2 samples per patient, not 1. Then there's an opportunity, perhaps with 2 images and finding a delta, to have also single subject studies in imaging. And then you get p-values and effect sizes at new types of signs or current signs, and then comparing groups of subjects is easier because you can do a lot of dimension reduction. You don't need to study something that's not significant in a patient. It's probably not relevant to the group. So when you collate patients together, you need something that's significant in at least 1 patient or 2, regardless of the group assignment, once you want to do a classifier. That's very important in machine learning. If you start with billions of features like we have in imaging, it's a very difficult problem to have a small cohort. Then you're creating a ginormous cohort, and maybe when we're a galactic civilization, we can study rare diseases in imaging. But otherwise, we need to change the paradigm. And really, the fourth industrial revolution would require to think of the hardware, the software that goes into imaging in the clinical trial as a system. And if the mathematics, just like CT scans and MRIs were invented, what if imaging is part of the clinical trial, and what are the type of adaptive statistics that are used there, other type of statistics, and how does that influence how the images are collated or analyzed or pre-analyzed beforehand, rather than waiting like the assembly line and saying, well, radiologists work there, and the clinical trialists are working here, and the statisticians work over there. I'll show you how that works in the genomic space. I'll just try to be conscious of time here. So I think I've already touched the fact that there are terminologies in medicine. There's a scalar theory that addresses that. The expert systems, I've discussed that, so we'll go rapidly here. This will just illustrate how we can operationalize learning about this pathway that's really, unless you measure the output, such as the bleeding time, it's really not measurable, right? The pathway function in the body, it's complicated except for the bleeding time. It's not exactly the pathway. So you can use annotation of molecules and pathways, genetic diseases that are disclosed in OMIM. And so you have the gene, and the gene can be associated to the pathway, and the disease can be associated to SNOMED. And then you can mine. SNOMED is a whole network of classification. Proliferative diabetic retinopathy is not assigned to an eye disease like an ICD. It's assigned to everything it is. It's actually a cranial disease, a cranial nerve disease. The retina is a part of the cranial nerve. It's an eye disease. It's a blood vessel disease. It's an endocrine disease, right? Diabetes. So you have all these classifications possible. We call these networks directed acyclic graph. So it's like multiple trees over it. You could systematically study thousands of classifications in medicine, tens of thousands, against tens of thousands of pathways. And we've run that, and you get a good p-value after control for false discovery rate, even though there's a lot of combinations there. And that comes out as the signal. And I've shown you the specifics that gives you the p-value for an overrepresentation of known Mendelian diseases that are in the hematological system and associated to a complement coagulation. And that gives you a systems property. And this system recapitulates thousands of book chapters of subspecialty, many of which any given subspecialist would not know about because it's studied in other parts of medicine, and new facts as well. or new knowledge. The way to think about it was presented mathematically by Marsden Blois, a physician informatician from UCSF, in the mid-1980s, sorry. And it's the scalar theory of biomedicine, where at each level you have an encapsulation of phenomena that occur at a lower level and new properties arise. I know I'm not showing you the maths, but that's what we're using. There are very few groups that leverage that, but this is what it would look like in terms of qualitatively, and how you discover an intermediate phenotype, the coagulation pathway from the molecular causes that you can measure, and the clinical aspect, like the coagulation time. We're not the only one to say that. There's a New England Journal of Medicine paper in 2007 that was asking to look into the intermediate pathway phenotypes for transcriptome classifiers, because the results were nonsensical. There were 10 classifiers published in that New England Journal for breast cancer metastases, and these were not overlapping at the genetic level. So mechanistically, it made no sense. We've published the papers that showed how they were related through protein interactions and networks. So coming back to learning from machine learning in omics, and I'll be relatively fast here. I've already told you about the problem of the fact we're not treating enough diseases. But how do you get a p-value and an effect size from one observation in each condition, right? That's statistically a problem. Conventional wisdom would tell you, you try to calculate the dispersion or variance between subjects, right? And now you're within a subject. Well, clinicians and biologists, they can make diagnosis with even incomplete facts, false facts, right? A false positive. And we do that all the time. We have a frame of collection of data, and not every diabetic has everything in that frame. Not every hypertensive has everything. So can we take external knowledge that frames the knowledge about pathway here, or about radiology and imaging at a certain scale of these neural networks? Remember, it's a bit more complicated. Here I can work directly with the crisp data. You have to do some transformation in imaging before you get to that level, and change the paradigm of case controls that you see to the left to a single case control in one patient, which is now isogenic. And think of the power we have with cellular and animal models. We bring that back to human beings. And then afterwards, we have very few features, only those that are significant in at least one person. And then when you look at other subjects, you get better power. We can obtain this knowledge externally from these knowledge bases I've told you before. There's many more here I'm showing. Unfortunately, I did try to annotate in SNOMED the radiologic signs and symptoms in 1994. I still remember 5,000 of them. And the board refused to take them. I was on the board later in 1998. So it's still not in SNOMED. I'm telling you, warning, there's a lot of grunt work. I did not enjoy doing that. It's a lot of hard work, but I wanted an AI in the electronic record we're designing. We did also identify that 20,000 signs and symptoms were in it from clinical medicine, and 5,000 were missing. Again, very hard work. I can tell you it's like several $1 million of cost to identify these things and organize them. It cost $20 million to transform SNOMED into an electronic form by the National Library of Medicine, paid to the College of Pathology. When I was on the board, we obtained that. So the motivation is really, in our case, in the omics space, is to analyze this bottom part here, the dynamic part of the genome, because a reference transcriptome or metabolome doesn't make any sense, because it's about other people, and actually someone that's living in the Arctic may be totally adapted with a transcriptome that would look aberrant in a reference transcriptome made from continental US people, right? So using one's own transcriptome, we're using less restrictive assumption, because we'll be looking into pathway level dispersion. So the dispersion will be within a frame of collection of data for one patient, but not the entire set. So there's subsets that are meaningful together for certain things. So I'll go more rapidly here. So there are other people that try to leverage genome by environment interaction, as we're doing, but their statistics were requiring more subjects per group. And the second and third row works only in isogenic studies of animals or human, but they still require 3 to 5 samples, while we're proposing only 1 per patient. This is how it works. We take transcriptome, let's say of a healthy tissue or cancer tissue, or an ex vivo assay is done, and we have aliquoted the blood separately with a control and exposure, for example, to coccidioidomycosis for valley fever, a grant we have. And then we calculate the transcriptome, and for each pathway we take, on the top, we take the subset, and we can calculate, there are 4 different statistics we've developed that work. It could be clustering on the fold change, looking for abnormal genes and normal clusters, or mixture models. How many of you are familiar with the statistics of mixture models? Pretty elegant. It's looking to these 2 normal curves that are there and assigning it to one or the other. And then you do an overrepresentation in the pathway, and you now have an effect size and a dispersion that are described at the bottom for each patient in each pathway. This is just to illustrate, this was an award-winning paper about 8 years ago when we presented these new technologies of the biological processes that are significantly altered in patients, of 55 patients of TCGA, the cancer atlas for the lung cancer. And you have the count of patients on the bottom for each pathway. And I'd like to point out this area here, which correspond to the immune response, where you have 5 to 10 patients per pathway. And you have many other pathways here of immune response where you have 20 patients. Now, you're all familiar with the biologics used in cancer now that are generally inhibitors of some of the immune processes, right? They're antibodies targeting that. They're targeting these processes because these are amenable to analysis by conventional statistics. Look at the larger number of uniquely altered patterns in fewer patients here. That's an example of new targets that are unveiled by these statistics, and it's an example of going beyond the 2,000 diseases we're treating, right, to the next set. Because now the diseases will be microstratified by their mechanism. And I don't think there is one type of adult onset diabetes. I don't think there's one type of hypertension either. There are probably thousands of types, so it's actually probably that bad. This is what it looks like when you do an analysis to compare in a meta-analysis the single subject studies. These are the subjects that are disease-free after 5 years, and those that died of disease. When a pathway, each line is a different pathway. For example, you have homeostasis, chromosomal localization, and so on. And when it's abrogated, you see the gray zone. When it's overexpressed, you see in green. You can see the patterns are quite obviously the same at the bottom and the top. Now, in 30 seconds, you can now read the dynamics of a transcriptome in a single patient faster than learning an EKG. And I would challenge anyone that the transcriptome of an entire body is much more complicated than an EKG, than the heart function. That's informatics, right? Bringing information in such a way that humans can consume it rapidly and understand what's going on. Now, I won't go to I'll just show you another study here. Well, I'll show you another study here. That's a clinical trial in which we've took 20 patients to design a classifier that could classify pediatric asthmatic patients that would be hospitalized or not in the coming year. They were all clinically equivalent. There's no difference of age, gender, ancestry, pulmonary function, or treatment between the group that will be hospitalized, the 11 that will be hospitalized, and the 10 that won't. Now, the peripheral blood was taken at the beginning of the study, exposed to rhinovirus, which is now known to cause 70% of the hospitalizations for severe asthmatic patients. They were all severely asthmatic, and the lung provocation assay was equivalently bad in everyone and not differentiable between the two groups. Well, they were not separated, right? At the onset, you have 21 patients. This is a fully specified classifier, and I'll show you the output. I've trained you how to read an output, right? Now I've put each patient into a contingency table. That's probably the first time you see that in your lifetime. Each patient is now clinically interpretable for what's going on and it's wrong. Is it the acquired immunity? Is it the innate immunity? That's what was discovered in the first cohort and validated in the second one. And you can rapidly see what's going on that's different, right? So there's abrogation of these areas in the true negative, those that are net hospitalized, and that would be the orange and yellow, which is the acquired immunity is abrogated and the chromatin organization. And there's overexpression of these two on the top. And this is a very small trial, and we had a sample of convenience to train the classifier. I won't describe it because I'm running out of time, but it's already significant. That was a proof of concept. Conventional transcriptome classifiers take 250 patients to train and 250 to validate. You're in presence of 20 patients to train and 20 to validate. A dimension reduction of tenfold. Clinical trials that could work in rare disease, that are using intermediate phenotypes or molecular phenotypes or pathways would be amenable to this. I propose that in imaging, the same thing is possible. You could have a wheel like this, in which you have radiologic signs, those that you already know from the 5,000 plus that the community has designed, and probably several thousand, tens of thousands more learned by machine learning on large corpus, which would be interpretable by human beings. Look, you have the name of what's going on here. You can read it. And you have a representation that's very simple. And you'd have your groups of radiologic signs that predict something that's meaningful. But again, you have to rethink your studies. It takes 2 measures of imaging in each subject. How many clinical studies do that? You have time series, right? Sometimes, but sometimes you have only one radiology. So you have to rethink clinical trials with clinical trials. There are other ways. These are circulating cancer cells. We've applied it in many different fields, and it's working in a lot of places. So I think I'll stop here because I'm running out of time. And lesson learned is let's try to leverage these new machine learning algorithms to learn intermediate phenotypes that are meaningful to human beings and are interpretable, rather than just the black boxes we're seeing right now. And to do that, there are strategies that were well designed, that have worked in the past, that could be applicable. It's not without sweat and blood, right? I didn't enjoy creating ontologies. It's a labor of love and blood, but it had to be done. You know your clinical signs, your radiologic signs. Unfortunately, I don't think they are currently structured. It's not just a data dictionary of listing them, right? You have to structure them as to what they're measuring, to the anatomy, to the morphology. It's a network of relationship. These 350,000 concepts of SNOMED have over 10 million of types of relationships, right, between them. And some are partami, some are association to the anatomical site or to the abnormal function or to the abnormal process, but clearly all of that can be done. It's doable. And the opportunity is really to dream and execute the future you want, rather than being dictated by corporations that only have a $ sign ahead. And imagine the future you want to realize, and you're all actors in this, and there's really a huge opportunity. This is the time, right? How many times in our lifetime can we be part of a new revolution of a market? And it's occurring right now, under our feet right now, if we're not participating. I started programming at age 13 on mainframes. Microcomputers didn't exist, right? But I did write at age 18. I created my first company, and the IBM PC came out when I was 19 with an open architecture. Before that, the Apple and the Commodore were closed architecture. It didn't create the market. The market exploded with the IBM PC, and I actually worked for IBM at that time as well. So I could see that opportunity of a revolution. The revolutions are occurring more rapidly. It's not just the end of the digital revolution with machine learning. You know better in radiology. You've been part of the 4th industrial revolution since 1985. With the exception of radiation oncologists, I don't think there's another specialty in medicine that has benefited from the wisdom of being part of changing the hardware, like the sensors, and the biology, and the mathematics at the same time, right? The computing. You had to do all 3. Your peers, especially the older peers, have like 35 years of experience in doing that. There's no other discipline in medicine but radiation oncology that has that kind of experience. So it's really on your shoulders to train other physicians into this revolution. You can lead. Thank you very much for your time.
Thank you for that. That was fascinating. Does anyone have any questions? We have some time. Yes.
Hi, thank you for the talk. So I was wondering on your temporal dynamics, when you looked at the transcriptomes through time, are you having to acquire a normal sample of tissue at each of those time points, or do you use back?
So that's a good point. I went very fast on one slide where there's actually another group that's very successful. It's Snyder. He's tested his transcriptome in urines and blood for the last, I think, 15 years every day. He used to be at Yale, now he's at Stanford. He's probably the $100 million man. He's not the $1 billion man yet. So they use time series, and actually in the last 5 years, time series were previously very difficult to use because you needed regular Cartesian time points that are always equally distant. But now there's new statistics that allow for flexibility there, which looks more like clinical practice. The statistics we're proposing is only 2 samples, so it's not necessarily time points. We've shown that it works with time points, such as HIV drugs within the first, so before giving the HIV drugs, and within the first week, obtaining a blood sample and verifying the dynamics of the transcriptome between these 2 points, we could predict 3 months later the CD4 increase. So we knew which ones would be resistant and which ones would not. No, that's useful because actually you could do remember how many times that clinicians have to start empirical treatment because they're not exactly sure what they're doing. In infectious disease, it often takes 2 days to get the antibiogram, right? So for the first 2 days, you're treating presumptively, you have the gram, and you say, oh, that's probably a strep, we'll treat along with penicillin, and then you learn, well, no, it's this other microbe, and it's resistant to this and that. So we could move away from being empirical. Tissues that are amenable to that would be, for ex vivo assays, would be the blood. I remind you all that 1/3 of the diseases are not only either infectious or autoimmune, but they're also immune-mediated. The example is asthma. So let's just take that example. If we know the biology of asthma, once a monocyte has decided to extravasate from the blood vessel and go to the lung and becomes a macrophage, its destiny is to never return to the lung, right? It will die there. So how is it that we can predict what's going on in the lung from the peripheral blood? Well, first of all, 50% of the genes are expressed in all cells in the body. So there's housekeeping genes and so on. So there's already a proxy. And they're really the ancestral cells. So ex vivo, we took something that's more primitive, that's more ancestral than the macrophage, and it was mixed with other white blood cells, but there was enough signal there on a genome scale basis to find the statistics and determine what's going on in the classifier. And by the way, these classifiers were meaningful, right? You could read it and it made sense for asthma. The innate immunity, the acquired immunity came out. We did not design that. There are thousands of pathways that were tested. Cell cycles were there. For cancer, that's what would pop up. So only 2 samples. It doesn't need to be at a time point. It could be an ex vivo assay as well. With the right tissue, so the skin is minimally invasive. When there's cancer biopsies, you can take the peripheral, the unaffected tissue at the periphery of the biopsy and the cancer itself. Any other questions? Yes, Bruce.
So your trials of N of 1 or N of 20 imply that maybe you have to have more precise phenotyping. And as imagers, of course, we're phenotyping the images all the time, but I guess it would imply maybe more precise phenotyping with imaging, maybe better structure reporting, and things like that. How can we help with the more precise phenotype?
So I'm not sure we need necessarily more precise phenotype. What we need is to learn explicitly and unveil the intermediate phenotypes upon which decisions are made by systems right now. And that can be mined, and it's not impossible to mine. I've shown you, again, don't tell my peers in precision medicine, but a simpler way to do it, right? Once you have a crisp signal with features that are comparable between patients, so you're not at the raw image anymore, but the system is at a scale where possibly microphenotypes and phenotypes at organs level. So is it the heart, the lung, when you have a chest x-ray and so on, is understood by the system. And these features now are in the same area of the matrix, because otherwise these neural networks won't learn it. Currently, they hide it from us, but there are ways to expose them. A single subject study was a way to expose it because we started with 25,000 transcriptome. But the dirty little secret is that, I didn't tell you, we decided that if it's that significant after false discovery rate 5% in one patient, and there were 2-dimension reduction, we're not studying transcript anymore, not 25,000 transcript, but 5,000 pathways, 5-fold reduction right there. And among the 5,000, any given patient had only 60 positive. And the union between the patients, that's not cheating because we're not looking into the phenotype, across all the phenotypes, was about 700 to 900 pathways. So we went from 25,000 features to 700 pathways. And then the machine learning algorithm could work with 20 people. When we tried to use conventional transcriptome and wide analyses, they all failed, right? We know it takes 200, 150 at minimum patients to do a classifier that way. In imaging, it's probably quite challenging as well. That's why these convolutional neural networks that feed into one another reduce the feature space. And you can see that because they actually are matrices. And you know how many inputs are coming from the previous neural network to the next one, and how many rows you have. And it gets smaller and smaller. And for those that include laboratories and clinical data, you can actually see that the laboratory is designed, there's an art to that, and the engineers that designed that in top journal papers, they include the laboratory before the clinical signs and symptoms or the disease output. So at different scales. But right now, it's like heuristics, and it's not formalized. And I would just propose it's much more elegant and strategic to use the same quantity of cascade of neural networks as you would have in scales of biology. That makes sense, not by the meter, but by the cellular anatomy, subcellular anatomy, tisular anatomy, and so on and so forth. And then you include the other phenotypes at the scale that make no more sense when you start looking into a classifier that has to learn from both imaging and clinical data. But there are techniques, there are people that are trying to do high throughput phenotyping as well, and that's yet another value, right? It's valuable, but it's not required. Right now, I think the crisis is that we don't understand how they're working in many cases. We've devoluted instead of evolving from what we knew from the 70s. Another question?
I have a question, Yves. I think this is very thought-provoking lecture, so thank you so much. So I try to just, understand what, you have been teaching us today is that, let's say, local biomarkers or biomarker is very short-range information. It's not succeeding, because we are still treating 4% of the disease and spending how much? $25 billion, and still we cannot actually come up with an effective treatment, right?
Yeah.
And the AI, yes, you can apply this to create the intermediate, range or long range, or actually even latent, relationship-based biomarker to help a clinical trial, which is wonderful. But I like to challenge a little bit though, because yes, AI is not the black box. It's just a computer algorithm. So once you understand the latent relationship by the AI or ML, really translate that to the treatment development, more effective treatment development, you clearly need a mechanistic research like more traditional. But if the AI comes up with much more latent information, not the single pathway, how are you going to translate that? I think this is going to be a major challenge.
So here's what I propose, Satoshi, and it's pretty straightforward. I didn't tell you, but like the GWAS have very low yield for human diseases. It's also a multi-billion-dollar study, the GWAS studies, and it was called the largest failed experiment in modern history, even by NIH, not Collins, but others. Because for diabetes, we now know the largest cohort was 40,000 people. We now know 270 features, right? They add up to less than 10% risk, and we know it's 85% inheritable. That's called the Inheritability Gap. because the model is simple, it's just linear. And it's not addressing more complex than a Mendelian model, and it's actually the wrong model. If you wanted to do the interaction by those statistics, you would need 20,000 times more patients. So 20,000 times 20,000, that's why you're speaking of galactic populations, right? When we're a galactic society, we may have enough diabetes to do that. But right now, we're not in the billions, and that's what it would take. What I didn't show here is another approach where we do dimension reduction with pathways at the genetic level, and we've confirmed many results that are wrong at 23andMe, for example, and published that with Jason Moore, that one of the authors of the papers you saw there, and the head of the All of Us study at the NIH, the million people being studied there before he was the head there. So the strategy is just do some kind of omics at the same level, probably not genetic, probably not the DNA, probably the transcriptome. And if you have the dynamic change of transcriptome in two conditions and the imaging, the intermediate phenotype for that, it's much more proximal, and here's why. In GWAS, the GWAS were very good in cellular toxicity. They're actually very predictive. So if you want, they're like, the effect size are 40% predictive, 60% predictive. It's all about scales of biology. The SNPs are at the nanometer. They're just one DNA molecule, right? And the cells are at 10 minus 5 meter, rather the organism, which is at the meter. So you've just already reduced by half the difficulty. So similarly, some of the intermediate phenotype that we're speaking of in radiology and your radiologic imaging signs are much more proximal to the omic space. And that's the way to leverage that. If you start predicting at the omic space what's predictive of these intermediate phenotypes, you now have the language and the association and network to start predicting how groups of molecules and groups of intermediate imaging phenotypes are predictive of therapeutic response, of disease progression. And that's a strategy I would approach. It's costly, right? But that would leverage what we know happen with cellular phenotypes and toxicity to drugs in GWAS. The GWAS worked there perfectly. I would claim that should work as well. We should write grants. Anyone ready to write a grant between the two groups? Thank you very much. I think I'm taking too much of your time now.